Welcome to The Explainer. We're about to tackle one of the biggest questions out there. How does life, build itself? All right, so today we're diving into a huge mystery in biology called morphogenesis. I mean, think about it. How does a single fertilized egg, just one cell, somehow organize itself into a complex creature? Whether that's a fruit fly, a mouse, or you. It's not just about getting bigger, right? It's about creating a very specific shape, a perfect form. So how on earth does it happen? So, to even begin to understand how an organism pulls this off, we've got to start with the instruction manual. And yep, that means we're looking at the genetic blueprint, its DNA. You know, we usually think of DNA as this static thing, right? Like a blueprint for a building. But the way scientists are looking at it now is totally different. It's not some fixed plan. It's way more like a dynamic program that's actively running inside every single cell, telling it what to do, what to become, and exactly when to do it. And at the very heart of this program is something called a gene regulatory network, or GRN. Now, don't think of it as a simple on-off switch. It's much more like a crazy, complex circuit board inside the cell, made of genes and proteins that are all controlling each other. So, how does a circuit like that actually work? Well, imagine a cell gets a signal. Could be from a neighbor cell or maybe just a chemical in its environment. That signal flips a switch on the GRN and boom, a specific gene gets activated. That gene then makes a protein, and that protein literally changes the cell's identity. It pushes it down a certain path, completely altering its future. And what you get from this process is what biologists call a differentiation tree. You start with just one parent stem cell, and from there, you get this whole cascade of branching decisions. The program just keeps running, and the cells start to specialize. They become what are called progenitors, and then boom, they turn into these highly specific cell types, like neurons that fire signals or muscle cells that contract. It's amazing. One cell becomes this huge diverse family, and every member has its own unique job. Okay, so the cells have their assignments. They know what they're supposed to become. But, that's really only one part of the puzzle, isn't it? I mean, just having a list of specialized parts isn't the same as actually building a house. How did these new cells physically arrange themselves into tissues, organs, and, the final shape of the organism? And this is where things get really cool. It turns out you need more than just a blueprint to build something. You need physics. The cells themselves aren't just the bricks and mortar. They're also the construction crew, actively using physical forces to shape the embryo. To pull this off, cells behave like a special kind of active matter. Think about sheets of cells, which are called epithelia. They can act almost like a liquid crystal, allowing tissues to flow and reshape themselves. But then, and this is the crazy part, they can suddenly switch and jam into a solid state, locking that new structure in place. It's like a material that can decide for itself when to be fluid and when to be solid. Wild, right? So scientists are now realizing that to really get what's going on in development, they have to think like engineers. They need to dissect the physical responses of cells to each other, all the pushing, pulling, and squeezing that actually sculpts the body. And listen, these forces are anything but random. They are a super precise controlled mechanical engine. This coordinated pushing and pulling is literally what drives the incredible folding of tissues that forms something like your spinal cord, or the delicate branching that creates your lungs. Yeah, it's biology, but it's also a total masterclass in soft matter physics. Okay, so we've got the genetic program, the software, and we've got the laws of physics, the hardware. But how do you put it all together to see if your theories are even right? How can you possibly watch the entire construction process from beginning to end? Well, the answer, it turns out, is in the digital world. Researchers are now building these amazing computational digital twins of embryos. These simulations let them run experiments, test out what-if scenarios, and tweak the rules of development in ways that would be just impossible with a real living thing. And one of the most powerful tools for this is called an agent-based model, or ABM. The idea is brilliant. In an ABM, every single cell is programmed as its own individual agent. Each agent gets its own set of rules based on everything we know about its genetics, that's the GRN, and its physics, like how it's supposed to push or pull on the cells next to it. And the process for building one of these is just incredible. First, you start with real microscope videos of an actual embryo developing. Then, you use AI to go in and identify and track every single cell. After that, you program each of those cells with its genetic and physical rules. And then, you just hit play and watch as this virtual embryo builds itself on a computer, and you can see if it matches up with what happens in real life. And this really just goes to show how far we've come. I mean, these models are so much more sophisticated than the older ways of doing things, like cellular automata, where cells were basically just pixels on a grid following super simple rules. In an agent-based model, each cell is its own agent with a complex adaptive strategy. It can behave much more like a real cell does in an embryo.
So, after all that, what does this actually mean for the future of biology? Where is this all heading?
Well, for one thing, we've learned that understanding how life builds itself is way more than just reading a genetic code from some static blueprint. It's about seeing that code as a live program in action.
And on the flip side, it's also not enough to just understand the physics, the mechanical forces, or the material properties of tissues all by themselves.
The really crucial takeaway here is that morphogenesis is a totally integrated system. The genetic program and the laws of physics aren't two separate things. They are deeply, deeply connected, working together in this constant feedback loop every single moment to build a living thing. So, the big idea, the main takeaway, is that to really get how life builds itself, we need a whole new kind of biology. A unified biology, one that combines genetics, physics, and computer science all together. You have to understand the code, the materials, and the simulation all at the same time. And you know, this whole integrated approach, it's leading us to a really profound question. By bringing all these fields together, are we actually on the verge of discovering the universal rules of biological construction, the fundamental laws of how any organism, here on Earth or maybe even somewhere else, built itself?
Welcome back to the deep dive. We've got quite a stack of research in front of us today, meeting transcripts, notes on artificial life, new algorithms. It's a lot. Our goal today is to really cut through that density and get to the core. We're diving deep into computational biology. Specifically, we're looking at the absolute cutting edge, how we simulate, engineer, and fundamentally understand life's most complex processes. Think embryonic development, cellular mechanics, all that good stuff. The sources you've pulled together, they really feel like a blueprint for where the lab is heading. Discussions on things like Lemunia, advanced microscopy, deep learning for cell fate. It can seem a bit overwhelming, right? But our job here is to synthesize these, well, pretty disparate fields into a single story for you.
Exactly. And that story, I think, is really about bridging a pretty significant gap, a conceptual gap. We want to understand how researchers are moving from these abstract mathematical rules, the physics of flow, diffusion, force band 2, to actual tangible biological outcomes. How do you get the spiral on a shell or a working organ? For the longest time, understanding development was like looking at a total black box. You put inputs in, something happened, you got an output. But now, with these computational tools, we're definitely moving it into more of a gray box scenario. We're starting to know many of the individual rules, the components, but the real challenge now is understanding the whole framework. How does it all link together?
Okay, so let's start right at the beginning, maybe the most fundamental level, the math behind biological patterns. We often hear about reaction-diffusion systems. Things like the Turing model, Gray-Scott equations. These basically show how simple chemicals, morphogens, right, diffuse and interact based on, well, fairly simple rules. And boom, you get spontaneous patterns, stripes, spots. It's kind of the classic explanation for, say, zebra stripes. Elegant.
It is elegant, But, life is often messier than just simple diffusion. What gets really fascinating, I think, is when you start adding active physical force into that mix. A lot of biological patterning forming organs, aligning cells, involves what we call active force systems. This means you've got molecular motors actually pushing and pulling or generating shear forces. They're actively working against that passive diffusion.
Okay, so why is that distinction so important? Active force versus passive diffusion?
Because it pushes us into the realm of non-equilibrium physics. It's not just about things settling down. A perfect example, think about eukaryotic cilia or flagella. The way they beat spontaneously in that coordinated way, that pattern in both space and time, the perfect timing of the beat. It doesn't just arise from a static concentration gradient. It needs energy, constant motion.
Got it. And if we shift gears a bit from those continuous equations, what about discrete systems, cellular to automata, CAs? You mentioned as a specific one, Lineadia. It sounds like it's generating some incredibly lifelike structures. What makes Lineadia special?
Lineadia is really interesting, partly because it feels like a philosophical bridge, almost. Yes, it makes more realistic artificial life than older CAs. It avoids problems like patterns just exploding uncontrollably. But the really key insight, mathematically, was realizing that any cellular automaton can actually be represented as a convolutional neural network, a CNN.
Wow, okay.
Yeah, So the very rules generating these complex artificial life forms, they have a structure that's fundamentally similar to the algorithms we use for, say, image recognition in deep learning. It directly connects artificial life research to the architecture of modern AI. That's a powerful link.
That is a stunning insight. Okay, so we have the patterns. Now, what about the stuff being patterned, the material itself? Our sources talk about jam solids and shear jamming. For our listeners, what does that mean when we talk about tissue elasticity?
Okay, think of a jam solid like maybe dense sand or even biological tissue itself. It can resist some forces, act solid, but flow under others like a liquid. It's right on the edge. Shear jamming is a specific type of phase transition in these materials. It's about stiffness, technically the shear modulus G. With shear jamming, that stiffness changes discontinuously. It suddenly jumps up when you apply a sideways, a shear force.
Discontinuously, not gradually.
Exactly. That's different from isotropic jamming, where stiffness kind of fades away smoothly. That abrupt jump is really key to understanding how soft biological materials handle mechanical loads and sudden stresses.
Right. And sticking with mechanics and movement. When modeling flows cells moving, fluids in an embryo, researchers use different viewpoints. Lagrangian versus Eulerian. Lagrangian, I gather, is like following one specific particle on its entire journey?
Precisely. You're tracking the individual. You follow that single cell or that little bit of fluid through space and time. You map its whole path.
Okay, so when is that useful? Why track the individual?
You'd use a Lagrangian approach when you really care about something like cell lineage, tracing a cell's ancestry, or understanding the specific mechanical history of one particular cell. Where did it start? What forces did it feel? Contrast that with the Eulerian view. There, you're like sitting on the riverbank. You pick a fixed spot in space and just record whatever flows past over time.
Right.
You use an Eulerian analysis when you're interested in the overall flow of, say, nutrients or signals through a specific region of tissue. You don't necessarily care which cell is carrying them, just the overall flux at that location.
So tracking one car versus measuring traffic at an intersection.
That's a great analogy. Yes, exactly. #tag2, decoding cellular architecture and development.
Okay, let's move inside the organism now to the building blocks, tissues. We need to appreciate just how complex tissue structure is. Take epithelial cells, the basic lining cells. We might picture them as neat, uniform bricks. But the sources really hammer home their heterogeneity. They vary a lot in size, shape, and even within a single layered epithelium, it's rarely uniform. The bottom layer might be cuboidal, short, and squat, while the top layer is tall and columnar.
And that diversity is actually crucial for the tissue's integrity, its function. To handle the constant mechanical stress that comes with these different shapes and packing, epithelial cells rely heavily on intermediate filaments, IFs. You know, we often think of actin as the dynamic part of the cytoskeleton, the cell's highway system. But IS, they're more like the steel rebar within the concrete. They turn over much, much more slowly. And they work together with actin to provide really critical, durable, load-bearing support.
And apparently, under extreme stretch, this IF network can actually reorganize into these star-shaped structures.
Yes, exactly. It's like a physical defense mechanism. The cell rearranges its internal scaffolding to better withstand the force. It's quite remarkable.
So understanding this architecture and mechanics is what lets us push into synthetic biology, right?
Absolutely. It's why we've been able to move beyond just simple organoids, which are small clumps of tissue. Now we have things like gastruloids. These can actually recapitulate gastrulation, that really early critical stage where the fundamental body layers form.
But the sources suggest the real frontier now is the embryoid. Just how sophisticated are these models getting? We hear about mouse models. What can they actually do?
Embryoids are significantly more advanced, yes. They can mimic key features of development after the embryo implants in the uterus, things like the bilaminar disc, the epiblast, the hypoblast. In human models, this can take us up to roughly day 13 or 14 post-fertilization equivalents. And the examples are, frankly, astonishing. Stem cell-derived mouse embryos grown inside an artificial yolk sac. They've been shown to develop anterior brain regions.
Wow.
And even a structure that resembles a beating heart.
A beating heart. That's incredible control. But is it, coordinated? Is it like a real embryonic heart or just kind of random twitching? How robust is it?
That is the million-dollar question, isn't it?
Yeah.
And getting that level of control, that coordination, it takes more than just adding the right chemicals, the right growth factors. This is where tissue mechanics and physical flows become absolutely essential. We're learning that physical flows like Marangoni flows, which are driven by differences in surface tension, like in soap films, can actually amplify tiny initial biochemical signals.
So the physics helps the chemistry.
Precisely. These flows can help establish the primary body axis, break symmetry in those early stem cell clumps.
Mm-hmm.
They physically organize the system so the chemical signals get interpreted correctly. It ties physics right back into development.
Okay, that makes sense.
And it scales up. If you need a whole sheet of cells to respond collectively to stress, maybe change shape together, or even split apart, individual cell responses aren't enough. You need plasticity at the tissue level. The individual cell responses have to be synchronized across the entire sheet. And that requires coordinating factors, signaling between neighbors, physical connections, making sure everyone moves in concert. Tag 3. The new computational toolkit for biology.
Right, tracking all this complexity, it obviously needs some serious computational power. Let's talk about cell fate. How does a cell decide what to become? Differentiation seems to be heavily regulated by epigenetics specifically, methylation marks on DNA, often clustered near genes. And the sources highlighted a really key finding from C. elegans, the worm. This leads to a mosaic pattern of differentiation. What does that mean, mosaic?
That mosaic pattern idea is crucial. It means that even if cells have the same genetic potential, they don't all switch into their final differentiated state at the exact same time. There's a temporal aspect that's staggering. Some start sooner, some later. It's not simultaneous.
Oh.
And understanding that timing, that mosaicism, is absolutely key if you want to accurately map out a cell's lineage path.
It kind of reminds me of that classic work by Darcy Thompson from way back, those amazing grid warp diagrams showing how different species shapes relate.
It does have that feel, doesn't it? We're essentially doing a modern dynamic version of that, but, with much more sophisticated math. Today, we're looking at morphological changes through things like dynamic network topology, geometric analysis. Thompson showed that shapes were related. Now, computational biology is trying to figure out why. And this leads directly to a major challenge mentioned in the sources, something called dimensional insufficiency in causality models.
Dimensional insufficiency, that sounds theoretical. What does it mean in practice?
In simple terms, it means if you try to model cause and effect just linearly, A causes B, B causes C, and so on, you often find your model doesn't have enough dimensions, enough variables to truly capture the biological reality. The real system, with all its feedback loops, its geometry, its interactions, it exists in a much higher dimensional space than your simple model.
So you're missing crucial factors.
Exactly. You're fundamentally under-modeling the complexity. And that's a huge hurdle if you want to move from just describing things to actually predicting them accurately.
Okay. Moving from theory to the practical tools needed, microscopy generates just vast amounts of image data. You can't track lineages if you can't even identify all the cells reliably. This is where AI tools like SAM, the segment anything model, come in.
Oh, SAM is a huge deal for microscopy. It's massively helpful for automating segmentation. Researchers can give it relatively simple prompts, just draw a rough box around a cell or scribble a mask, and the model does a remarkably good job of precisely outlining that individual cell, even in crowded images.
So it breaks the bottleneck of manually outlining thousands of cells.
Precisely. It's essential for building the large, clean data sets you absolutely need for any kind of large-scale lineage analysis.
Okay, so once you have those clean data sets, identifying all the cells, how do you analyze the relationships between them? That's where things like graph neural networks and TDA come in.
That's right. GNNs are suited for network data and topological data analysis, or TDA, using techniques like persistent homology. Well, it sounds complex, but the core idea is useful.
How does persistent homology help?
Think of your cells over time as a dynamic point cloud in space. Persistent homology helps you find the underlying structure, the topology, of that cloud as it changes. It looks for features like connections or loops between cells that persist over time, filtering out the transient noise. It helps identify the stable, meaningful connectivity within lineages, rather than just who happens to be neighbors at one instant.
And critically, when analyzing development, you have to use directed graphs, right? Not just connections, but parent-to-child connections.
Absolutely essential. Cell lineage is inherently directional. Mother cell divides into daughter cells. If you ignore that directionality, treat the network as symmetric, you lose fundamental information needed to predict fate. You can't mix up parent and child.
That makes sense. Okay, final area genomics and the big AI models. Large language models. We saw hyena DNA mentioned. It uses something called implicit convolutions.
Yeah.
What's the breakthrough there for genomics?
The big deal with models like hyena DNA is about efficiency and scale, really. They're managing to match the performance of the standard, very powerful deep learning approach, attention mechanisms, on really complex genomics tasks. But crucially, they can handle much, much longer sequences of DNA as input longer context lengths. And they do it faster with lower computational complexity.
So you can analyze bigger chunks of the genome more efficiently.
Exactly. It makes tackling massive genomic data sets computationally feasible in a way that was maybe hitting limits before. It opens up new possibilities.
That's a clear win for just raw processing power. But the final finding you pulled out was almost the opposite, that sometimes things are surprisingly simple.
Yes, isn't that interesting? Despite all this complexity we've discussed, researchers found that for some specific proteins, the relationship between the gene sequence and the protein's function is actually remarkably simple. Even when looking at a vast space of possible gene sequences, more than 10 to the power of 10 possibilities, they could accurately predict the protein's function using relatively simple, interpretable biophysical models.
So not everything needs a black box deep learning model. Sometimes the underlying physics is clear enough.
Precisely. It gives us these beautiful, understandable links directly from gene sequence to physical function. It's a reminder that sometimes elegance and simplicity still win out, even in biology. Hashtag tag outro.
Wow, okay, that was quite a journey. We started with relatively simple math describing patterns in nature, like Turing patterns. Then we moved through the complex mechanics and physics needed to understand how you might get something like a beating heart structure in a dish. And we finished with this, frankly, enormous computational toolkit AI for segmentation, graph networks, large language models that's now essential just to track lineage and try to predict cell fate.
Yeah, the whole trajectory is really about moving beyond just description. The goal now is predictive power and eventually design capabilities, what some call morphogenetic engineering. We're actively trying to build the fundamental frameworks to understand why life organizes itself the way it does, not just documenting that it does. And maybe, a final thought to leave with you, the listener. We talked today about how all these evolving biological systems are made of diverse parts, combining into complex, let's call them configurational states. Instead of just focusing on simple linear cause and effect, this gene causes that protein, which causes that behavior, perhaps the next huge leap forward in biology. Maybe it comes from truly understanding the sheer vastness of the possible ways cells could arrange themselves, all the possible configurations, and then figuring out the powerful physical and computational rules, the filters, that force life down the very narrow functional path that actually takes through that immense space. Why this configuration, and not countless others?
The constraints that shape life out of infinite possibility. Definitely something to ponder. Thank you for guiding us through that complex landscape, and thank you for joining us on this deep dive into the frontiers of computational biology.